Hello, my name is Rosa Trobajo, and I'm going to present some of our works on diatom metabarcoding of Mediterranean rivers. And I will put particular emphasis on the contribution of some species to the discrepancies of the two methods, the morphology and the metabarcoding that we have observed. We studied benthic diatoms of the Catalan rivers, and at 164 sites that they were sampled as part of the biomonitoring program of the Catalan Water Agency. And for these 164 sites, we had parallel samples. That means we have morphological counts as well as DNA reads from high-throughput sequencing. The barcode that we used was the short region of RBCL, and for these results, for this presentation, we grouped the DNA reads in OTUs. Though the studies that we are doing, we are using amplicon sequence variant. We are using genetic variants, which I think it's a very good improvement. And the overall aim of the study that I'm presenting was to study the applicability of diatom metabarcoding to assess the ecological status of the Catalan rivers. For this, we compare the biotic index using diatoms, the IPS, as well as the ecological status assignation derived from this, the IPS, obtained by both methods, the morphology and the metabarcoding. First, to comment that using morphological counts, the numbers of species recorded was much higher than using DNA reads. And that the numbers of species share, that they were identified by morphology and by metabarcoding, was about 30%. And one may say that, or may think that at first sight, this 27% is not very impressive, or it's not very good, but it's important to know that this 27% of share species account for the vast majority of the total bulk counted by the light microscopy, and also for the vast majority, almost 90%, of the total reads recorded by high-throughput sequencing. So for the Catalan rivers, we could say that almost all the most abundant and common species inhabiting the rivers can be detected by morphology and by DNA. So they can be studied by both methods or the method that we choose. In relation to the comparison of the two methods, the performance in relation to the IPS values and also the ecological status assignation, we found a remarkably good agreement. Not only was the relationship very close to 1-to-1, but also the inception was close to 0. However, in 10 out of the 164 samples that we studied, we observed degradation of the ecological status, in the sense that in these 10 samples, the ecological status, according to the morphological inventory, was good or high, and it was downgraded to moderate or lower when we used the DNA data. And we call these sites critical sites because their importance in the Water Framework Directive. As you know, this directive requires for all water bodies that do not reach the good or the high ecological status, requires to the water managers to take remedial actions with the economical and other implications. So the rest of my talk, I'm going to present the analysis that we did to investigate the contribution of some species to the IPS value, paying particular attention to the critical sites to see if we could understand the nature of these discrepancies. And what we did is to apply a sensitivity analysis by leaving one species out at a time, and to see how the IPS was modified. That is, what we looked, it was the difference between the IPS value when the entire diatom community was considered for a particular sample, and the IPS value of this sample once a particular species was left out. So for all the species and in all samples, we had a positive or a negative value. and this was indicating a positive or a negative contribution, or a zero contribution. That means that the species has no effect in the IPS. This graph summarized the positive or the negative contribution of the 35 most morphological species. However, we did this for all the species in all samples and for both approaches, for the morphological, for the light microscope counts, and for the DNA reads from metabarcoding. And looking at the critical sites to see if there was any kind of special or interesting in the contribution of the species. This graph shows for each species the numbers of critical sites. The critical sites are in the x-axis. So the numbers of critical sites for which each species are ranked to be the first, the second, the third, or the fourth most negatively contributors to the IPS. So in other words, this graph is giving an idea of which are the species that are most influential in lowering, in decreasing the IPS score. Likewise, this graph shows which species are most influential in increasing, in being positively contributors to the IPS value. And from the two figures, we can see that there is one species, that is Fistuliferous saprophylla, that is the species that was the most contributor to the discrepancies of the IPS values from the two methods, from the molecular and the morphological approaches. Because Fistuliferous saprophylla was the most negatively contributor in the molecular inventory. In the molecular inventory, in 8 out of the 10 of the critical sites, Fistuliferous saprophylla was or the first, or the second, or the third most negatively contributor to the IPS. While in the morphological counts, Fistuliferous saprophylla was one of the most negatively contributors, but only in one of the critical sites. So to try to find out the reasons of the discrepancies, we analyze, we look at the relative abundance of this species of Fistuliferous saprophylla obtained from the two methods, from the light microscopic counts and the high throughput sequencing reads. And what we saw is that overall there is a tendency of this species, the relative abundance of this species by molecular markers, to be underestimated in relation to the morphological counts. However, it was a group of samples that we observed the opposite tendency, in the sense that for this group of samples, Fistuliferous saprophylla was not recorded, was not observed in the light microscope, in the light microscopies samples, but the relative abundance recorded by the high throughput sequencing data was relatively high. And all the critical sites are in this group of samples. And the explanation that we give to this is because Fistuliferous saprophylla, it's a very small, weakly silified diatom. So it's very likely that during the oxidation process it gets destroyed. So that is the oxidation process is a process that we need to have to prepare the slides to be observed in the microscope. So if it's destroyed, we don't observe it. We cannot detect it in the morphology. And for those samples where the Fistuliferous saprophylla was not destroyed, there was a tendency, as I said at the beginning, to be underestimated using the molecular data. And the tendency was also observed in other species, like Anantidium minutissimum, but very clear in Niche inconspicua or in Planotidium frequentissimum. And this is because what they have in common, these three species together with Fistuliferous saprophylla, is that they are very small species, small cells species. And we know that RBCL copy number is related in some way related with the cell volume. Therefore, small cell species tend to be underestimated, the relative abundance tend to be underestimated by molecular data. And the opposite pattern was found in ulnaria ulna, but for the same reasons. Ulnaria ulna, it's a very big cell, therefore the relative abundance with molecular data is overestimated in relation to the observations done by the relative abundance detected by the morphology. So the take-home messages from this study of diatom metabarcoding of the Catalan rivers would be: the first is that overall we found a very good agreement between IPS values and ecological status assessments calculated by the both methods, the morphology and the metabarcoding. However, in spite of these promising results, there are some mismatches and biases that need to be understood and addressed before a complete switchover to the DNA-based approaches are taken. And that for some of the species, the biases may not be relevant, since they may have no or very little effect in the final IPS value ecological status assessment. So it's very important to identify which are the species, the key species that drive the ecological status assessment. And for this, our sensitivity analysis has proved to be useful, at the same time that very straightforward. And also it's very important to have in mind that mismatches between IPS score from microscopy and metabarcoding are expected, because indeed we are analyzing the same thing with two methods, and that not always mean that one or another is wrong. For example, morphology-based assessments are based on cell counts, no matter is the cell size. For practical reasons, by microscopy, we agreed that we count one cell, whatever is the size of these cells. While metabarcoding is based on DNA reads, which are partly dependent on the cell size. So the cell size is already incorporated in the DNA reads. And until here is my presentation, so thank you very much for your attention. And just mention that in case you are interested in our latest research on diatom metabarcoding and the genetic variants, we are applying amplicon sequence variant. Javier Pérez Burillo is going to give a flash talk later in the poster session of today.